Hi, David. Thank you so much for joining me on this conference call today to celebrate World Microbiome Day. And I know that the microbiome is a subject close to your heart.
Yeah, it is. I mean, I've been in microbiology for many, many years, though my main role now is as a Workstream Leader in Biomarkers at Waltham.
So today we're going to talk about a recent paper you've had published about the dog microbiome, published in the journal Applied and Environmental Microbiology. And this was a really interesting study. I know it was a collaboration between you and other researchers at the Waltham Petcare Science Institute. So can you tell me a bit more about the research?
Yeah, so the background of it is it was really a nutrition and biomarker study. And at the time, I thought that it was such an unusual study to be doing that we ought to involve the microbiome team. And so I asked Kevin Hughes if he would be involved and lead a project on looking at the fecal microbiome in these dogs in this study, with this unusual diet.
So this was a study on healthy adult dogs. Tell me about, they started having, they were being fed a diet that was a standard, complete dog food, a bit like the one you would just buy for your dog. But then they were moved on to a really quite unusual diet, weren't they? Can you tell me why was that diet so unusual?
Yeah, so, well, it was unusual because what we needed to do was address the questions in terms of the nutrition aspects that the project was really about. And so what we needed to do was make sure that the diet didn't cause any confounding parts to the study. So we didn't want to have any complex vegetable matter or complex proteins. We needed to have purified amino acids, and we needed to make sure that whilst it was nutritionally complete and safe to eat, we wanted it to be really, really, really simple. So we wouldn't see any compounding factors for the study. So what that meant is that we were in a really weird situation of giving dogs something that we would never, ever give them again. And that's why we thought it was a really good opportunity for microbiome to be involved and have a look at how diet could have an impact on the microbiome.
So that sounds really interesting. So this diet had everything that the dogs needed to be healthy, but almost the nutrients, but without the ingredients that we'd not, there were sort of nutrients separated from the normal ingredients that we'd expect to see in dog food. So that must, yeah, I can imagine why that was a really interesting thing to study. And during this study, you analysed the DNA in the bacteria, in their feces, in their poo.
Yeah, that's correct. So one way in which you can get an understanding of who's there in terms of the bacteria that are in the gut is to look at their DNA, and that can tell you who's there. It can also tell you, if you look at it in a different way, what are they also able to do when they're there. But in this case, we're looking at who's there, and it's a surrogate. Got to be really careful that when we look at the feces, what we're doing is taking something that we can just pick up off the floor, if you like, and have a look at without needing to do anything invasive. A way of me describing it that might make sense to someone is if I wanted to know what happens in a town, I could go in and knock on every door and ask them what the names are and what do they do. But what I could also do, which might be a lot cheaper and a lot quicker, is to run a poll in the high street and just ask as many people as I possibly can, what's your name and what do you do? And that can, if you do it in the right way, and you know the pollsters make a lot of money out of doing this kind of thing. If you do it right, you can get a really good estimate of what the community is like, and that's kind of what we're doing. And we're trying to work out what the community of the gut looks like by the estimates from the feces.
And so that's what the word microbiome, when we're talking about the microbiome, it's that community, right? It's that community of bacteria and other microorganisms. The way you're talking about a community in a town, this is the community of bacteria inside the dog, basically.
Yeah, and we've got to be, it's interesting because when we talk about the community, it's not just about who's there, but what are they doing, or even what are they capable of doing. So there's a lot of elements of the microbiome which can be kind of overlooked by using such a simple term to talk about lots of complex areas. But in this case, just making it simple, we're going to be talking about who's there.
Okay, so who's there in terms of the bacteria, but also, like you say, it's not about just who they are, but like what's their job, or what could their job be in this sort of society inside the dog. So these dogs, I think there were 46 of them, right? They were healthy adult Labradors. Great choice. I've got 2 Labradors, lovely dogs. They were given this, they were switched onto this purified diet, this unusual diet, for quite a long time, for about 9 months. And what did you see when you were measuring or analysing this bacteria in the faeces?
Yeah, so we took our first samples 4 weeks after they changed diets, and then we took them throughout. And what we saw, I should say, what we thought we might see, maybe that would help as well. So what I was expecting is, because the diet was so simple, and there wasn't a lot of interesting things that bacteria could kind of get their teeth into, that I thought it would become quite a bland kind of community. I didn't think that there'd be many particular organisms there. So I thought the diversity might drop down. And I also thought that the kind of work that they could do would also drop. And so what we did find actually was that the diversity was about the same. So we saw the same different types of bacteria, if you like, but they were, what was interesting was that they were entirely different. So the community structure was really complex, but the people, the bacteria that represented it were very different under the 2 conditions that we looked at.
Okay. So I guess that's science for you, isn't it? Sometimes what you expect to happen does happen, and sometimes you get a surprise, you weren't expecting, which is why it's always so interesting. But what happened after this 9 months on this purified diet? They went back to their original sort of more standard dog food, and then what happened to those bacteria?
Yeah, so again, if I say where we thought it might have been going, so we did them together. Once they'd gone down to such a small community that we had imagined, it might mean that when the food came back on, if you open up the tap again, could there be a different bacteria that exploit that opportunity, because it wasn't a stable bacterial population as it was before. But instead what we saw was that it basically went back to where it was before. So the implication of that is that the gut microbiome, the actual bacteria that are in the gut, were stable all the time. And that, if you like, the only thing that was making the difference that take place in the fecal microbiome was which bacteria could exploit that diet under those circumstances.
Okay, so it sounds like the fecal microbiome, and maybe therefore the gut microbiome, because you mentioned this is a good non-invasive way of trying to kind of measure what's going on in the gut. It can change, or quite rapidly, sort of back and forth.
Yeah, and I think that it's really important, because it's got good news and bad news, if you like. So the good news is that it shows that the diet can have a profound effect on the fecal microbiome, and that means it might have a profound effect on the activities that they perform whilst they're in the gut. So what do they produce that the host can then use? Okay, but the bad news is that you can't necessarily say that what you see in the fecal microbiome is a true representation of what's happening inside the gut.
Sure. Yeah, I suppose it's just the kind of best the best window you have,
Absolutely,
if you like, into the inside world, don't you? So if dogs do have a gut microbiome that's that's kind of quite stable, it sounds like it might be quite important to set them up to have a healthy gut microbiome, at an early age, maybe when they're a puppy or first weaned onto, off their mother.
That's correct. Yeah. So what we really showed is that in healthy adult dogs, they have a stable microbiome, gut microbiome probably, and that does imply that if you want to have a beneficial effect on the gut microbiome, the best thing to do is to be involved in the development of that. And we know this is true in the case of babies as well, in humans as well, where depending on the type of milk that they're fed, or what they're weaned, how they're weaned, when they're weaned, these can be important implications for their health in later life. So yeah, while we didn't do it here, I think the work that we've done shows the importance of focusing on the early development of the microbiome.
Yeah, that does sound really interesting. And this study, all these dogs were, you mentioned, healthy adult dogs. So can we learn anything from this study for those dogs who perhaps have got different disease conditions, or maybe haven't got a healthy gut microbiome?
That's a good question, and really we can't extrapolate. I'm not saying that I can imagine situations where some of these dogs may benefit from having a diet for a short period of time, that can stabilize them and get them back to maybe where they were if they'd had an unstable dysbiosis, if you like, a diarrhea type thing. But it doesn't necessarily mean it's true for all dogs. There may be some persistent chronic cases where you would have to maintain feeding a good diet for a long period of time, or you may have to consider other interventions of a non-dietary nature to help support them and get better.
The more we talk about this, the more I realise this is such an interesting area of science, and there are still so many questions to be answered. So is this an area that Waltham researchers are continuing to explore?
Yeah, we've got a great team, really bright people, lots of enthusiasm and lots of questions that they are driven to find answers for. Yeah.
Fabulous. Oh, thank you so much, David. That was really great. Thank you for talking me through that really fabulous study, and have a happy World Microbiome Day.
Thank you for asking the questions, and the same to you too. Take care.